Good morning and good afternoon, everyone. My name is Mahalea Marshall, and I'm with the IQConsortium Secretariat. I'd like to thank you for joining the last of this 3-session webinar series on human mass balance. This webinar series has been presented by the hADME Working Group and is sponsored by the IQ Translational and ADME Sciences Leadership Group. I'd like to turn things over now to our moderator, Jason Bohr.
Thank you, Mahalea. Apologize, my camera switched on me. Today, I'd like to thank Cyrus Kajastan. I'd like to thank everybody for joining us today. Today's session is brought to you by the IQConsortium Human Mass Balance Working Group. Before we begin, I'd like to thank our former co-chair, Graham Young, and also our sponsors. Next slide, please. The working group was formed to discuss the conventional and historical C14 ADME study design and opportunities to adopt modern alternatives, along with discussions around the impact of the data produced. The working group was launched in August of 2023, and is targeted to sunset in the first half of this year. The working group consists of 4 subteams: regulatory requirements, technical aspects, clinical designs, and data impact. The output of these subteams garnered enough interest from the FDA such that we were granted the opportunity to present at their internal lunch and learn sessions in later of last year. And this is the third and last of our series of 3 webinars. Today, we will be discussing new and emerging technologies. Philip Kuykens will discuss quantitative technologies. Alex James will go through AMS. And Greg Walker will discuss fluorine-19 NMR. Next slide, please. Oop. There we go, thank you. We'd just like to acknowledge and thank all of the members of the working group for their time and dedication, and to point out that the ideas shared today are those of the working group and of the presenters. That's it, thank you. And now I'd like to turn it over to Philip.
Thank you, Jason. So the topic of today is quantitative technologies used in human ADME studies, where we'll first give you a brief overview of quantitative technologies that can be used in human ADME studies, and then we will dive into 2 specific technologies in more detail. So first of all, Alex will go over Accelerator mass spectrometry, and then Greg will give more insight into the technique of fluorine NMR for human mass balance use. So first of all, it must be said that still the most commonly applied technique today is still traditional radioactive detection. It's still called the golden standard in human ADME studies, where we typically label a compound with radiocarbon. In exceptional cases, tritium can also be used. Tritium is usually easier, let's say, to synthesize, but it suffers more from it's more likely that there is biological instability, and that's why radiocarbon is usually preferred for human mass balance study. Now, the nice thing of radio-labeled work is that you have a very selective detection, and you can easily discriminate your drug-related material from the background, and you can also even quantify metabolites where you don't even know the structure, because you get a structure-independent response. And on top of that, you can also make up a mass balance, because what goes in should come out. So if you dose a certain amount of radioactivity, you also know that you should ideally retrieve the majority of that amount back in excreta over time. Now, one of the main reasons why traditional scintillation counting is still the most commonly applied technology is that it's well-established technology. It's relatively cheap and also fast. But of course, there's one drawback, is that it relies on a decay, a beta radiation to be detected, and with a half-life of 5,730 years for 14C, it's not the most efficient and sensitive technology. And that's why accelerated mass spectrometry, where you detect individual 14C atoms based on the isotope ratios, is way more sensitive. And there, Alex will give more detail on the technology in the next part. But I briefly want to highlight another technology which is quite promising, and that is cavity ring-down spectroscopy, which is a laser-based technology where you first combust your sample and oxidize it to CO2. similar to the front end of an AMS system, as Alex will explain in more detail in a minute. And that CO2 is then transferred to a ring cavity. It's a kind of box containing 3 mirrors. And then a laser light is injected, a pulse of a laser light is injected that is traveling in between this, back and forth between these mirrors, so that a kilometers long flight path is created. While traveling, that light is traveling, it's absorbing the partially absorbed by the 14 CO2 molecules. And it's not actually the absorption that is measured, but more the decay. So the light will faint over time, and the decay, also called ring down time, will be measured with a blank sample, a reference sample, and a 14C containing, so your analyte sample. And the difference between this decay is then proportional to the amount of 14C in your sample. Now, cavity ring down spectroscopy is not commercial yet and not yet used in human mass balance study, but it's quite promising because it's very sensitive. There are attempts to improve it and to commercialize it, and there are already quite some results available in literature showing that it's, let's say, giving similar results to AMS, as illustrated, for instance, here on the left-hand side in a publication showing the correlation between AMS and CRDS for a small molecule, and on the right-hand side, similar correlation for an antibody. Now, of course, in the ideal scenario, we would like to see, to be able to do a mass balance study without a radio-labeled compound, so a cold study. And for that, we would need then a technique that is quantitative and giving a structure-independent response and able to detect and quantify elements that are, let's say, prevalent in drug therapeutics, but not omnipresent in the endogenous background, so that you can discriminate your drug-related material from the endogenous background. And there are 2 promising techniques that actually already were used in a human mass balance study. And first of all, that is fluorine NMR, where Greg will give more detail on in the last part of this IQ webinar. And a second technique is inductively coupled plasma mass spectrometry, also element-specific detection technique, where you first ablate a solid sample or evaporate a liquid sample into a plasma. It's an argon plasma of around 7,500 Kelvin. In that plasma, molecules will decompose into their constituting atoms, and then these atoms are separated and detected in a mass spectrometer. And since you're analyzing atoms rather than molecules, you also get a structure-independent response. And here you see an illustration of that, the use of that technique. On the left-hand side, you see on top in the black trace a bromine chromatogram, so bromine quantification for a compound, an anti-tuberculosis compound. And on the bottom, you see for the same dog feces metabolism sample, the analysis for the same sample by radioactive detection. You can nicely see the correspondence between the icp-ms bromatogram and the radio chromatogram. And that was a validation used to finally use that technique also in a human mass balance study for that anti-tuberculosis compound, where you see on the right-hand side an illustration of one of the feces samples from a patient sample, because that's also the benefit of this technology, that you don't need a specific separate human mass balance study in healthy volunteers, but you can just pick samples from ongoing studies. So here it was a patient sample, where in this case the bromatogram was created. And of course, bromine is not the most often encountered element in therapeutic drugs, but icp-ms can also be used for metals, for instance, iodine, chlorine, and other elements. So there's quite some promise, and I'm sure we will see more of this in the future. And with that, I give the word to Alex, who will dive into Accelerator mass spectrometry.
Okay, thank you. So what is AMS? So AMS is used to detect and quantify the amount of radioactive carbon in a biological sample. So the sample introduction process produces ions, which are negatively charged, and the samples are introduced into the ion source either as graphite or carbon dioxide. And the fact that these are negatively charged ions prevents confusion of the 14C signal with that of 14 nitrogen, as the latter does not form negative ions. So then the ions then pass through a magnet and an accelerator unit. The purpose of this is to select the ions of interest, of course, and to remove interferences, similar to what a mass spectrometer does, actually. And the ions of interest are then detected. And at the end, two key ratios are calculated, and that's the 14C-13C ratio and the 13C-12C, from which the 14C-12C ratio can be derived as shown. So then from that, then the radioactivity value in the dpms per milligram of carbon can be calculated, which is then converted to the dpm per milligram of the selected sample based on the total carbon content of that sample. Next slide, please. So from the point of view of AMS being used for carbon dating or biomedical applications, carbon-14 is the element of interest. Of course, carbon is not a monoisotopic element, but it consists of three isotopes: 12C, which is the most abundant one, dominant one, 13C, which is present at an abundance of approximately 1.1%, and 14C, which is present at trace levels, very, very low levels, as you can see in the center there. So carbon-14 is produced in the environment, mainly by the interaction of cosmic radiation in the upper atmosphere, from where it enters the carbon cycle, as you can see on that illustration on the left side of the slide. So living organisms kind of have an equilibrium with the environment. So when an organism dies and is preserved, the 14C contained within it starts to decay relative to the environmental background, which is the basis behind radiocarbon dating, which is what AMS was initially mainly used for when it was invented 40 or so years ago. So what actually is AMS measuring? So here I'd like to introduce the concept of fraction modeling, which you can see on the top right of the slide, which is basically defined as the 14C-12C ratio in a sample divided by the same ratio for the environment. And that chart you see on the top there is just an example of the fraction modern determination in Earth's atmosphere since 1900 to the present. The peak you see there was starting at around 950, was caused by the atmospheric testing of atomic weapons. And of course, after this practice was banned, the fraction modern in the atmosphere is now slowly returning to previous background levels. So for biomedical applications, since we are adding 14C artificially, the fraction modern value will always be greater than 1, whereas for carbon dating applications, this will be less than 1. So from fraction modern measurements, the radioactivity amounts in a sample can be calculated, considering additional parameters such as the carbon content of the sample and the radioactive background levels, etc. Next slide, please. So how are samples introduced into the AMS? So there are two methods, graphitization and combustion. And I say modern AMS instruments seem to be going away from combustion and towards direct introduction of CO2 generated from combustion of the sample. So how does this combustion process work? So the sample to be measured is weighed accurately into tin cups. And these tin cups are then burnt in an elemental analyzer, which separates and quantifies the released gases, in particular carbon dioxide, which is needed for the total carbon content of the sample, and nitrogen. And the CO2 is then trapped and released into the AMS, where fraction modern is made, the 14C-12C ratio and fraction modern is determined, and from that, the radioactivity content of the sample. Next slide, please. So why combustion AMS over graphitization AMS? So I think for biomedical applications, the main reason is that sample preparation for analysis is much faster. So basically, this fractionation of direct sample weighing into tin cups, followed by loading into the elemental analyzer, can basically lead up to a maximum of around 6 minutes analysis time per sample. Whereas for graphitization AMS, for example, an AEG3 graphitization system can only graphitize 21 samples over a working day, and that process is also very labor intensive. So 1 FTE is needed to prepare those 24 samples. However, graphitization AMS does have certain advantages. So there's a lower crosstalk or background with this instrument, and the main disadvantage for combustion is that more analytical run checks are needed, such as the performance of the elemental analyzer. But despite these things, it seems that for our kind of work, human ADME work, combustion AMS systems are the dominant one used these days. Next slide, please. So this slide just illustrates how AMS technology has evolved over the last 40 years. As you can see on the top left, they were huge instruments initially. They required, they were taking up a warehouse. So the size has dramatically gone down. On the far right of the far right image is a picture of the layer AMS system, which is made by a company called Iron Plus. This requires about 5 to 6 square meters of laboratory space. So there's a huge reduction in size and, of course, a huge reduction in cost as well, not just of the instrument itself, but also operational costs. In parallel to that, the user friendliness of the instrumentation has increased, and software too is more compliant. So the layer software is, for example, amenable to be for GXP analysis. And of course, in terms of sample preparation and throughput, so that with the combustion sources, sample preparation time and sample analysis time has greatly decreased, and with corresponding increases, of course, in throughput. And of course, also the combustion systems can be automated to a certain extent. Next slide, please. So one important message I'd like you all to take away from this is that AMS can be used interchangeably with other classic radio detection techniques, such as liquid scintillation counting. As you can see from that graph on the bottom, this basically compares a series of 3 radio-labeled compounds with different radio labels, one with 2 14Cs, one with 1, and one with 6 distributed around an aromatic ring. Independent of the technology used, you can see here liquid scintillation counting, low-level counting, and combustion AMS. You have a linear, basically linear line there. So so you can use them, and so of course the microtracer human ADME study, which I'll come to on my next slide, is one of the main things that AMS is required for. But however, AMS can also be used in support of classic studies in situations such as when quantification of total radioactivity in blood and plasma for long half-life drugs is needed when the concentrations drop below the LOQ of LSC, which would enable better characterization of the pharmacokinetics. Also, investigation of metabolite profiles at late time points where radioactivity levels have dropped below the levels measurable by liquid chromatography fractionation with top count. And so one can have a study where both technologies are used interchangeably. Next slide, please. But of course, the main application of AMS is to support microtracer human ADME studies. So first, a couple of definitions. So a microtracer refers to the amount of radioactivity contained within the drug product. So this is typically less than or equal to 1 microcurie. Microdose refers to the amount of drug product administered. So this is typically less than or equal to 100 micrograms administered IV, but also sometimes PO. Of course, then the radio-labeled drug substance and drug product is basically a drug substance drug product containing a mixture of unlabeled drug and drug labeled with a radioisotope. I've put the stable labeled definition on the bottom left, although it's not relevant here, but it's just to complete the set of definitions that you commonly see. So the most common study design options for microtracer studies that you see is just similar to a classic human ADME, so a single oral 14C microtracer instead of a higher dose to 6 healthy volunteers or patients. Or a single oral 14C microtracer dose to 6 healthy volunteers or patients, followed by an IV microdose together with an unlabeled oral dose after a suitable washout period. And this is a study I'll talk about on the next slide. And of course, very occasionally, but quite rarely, you see these absolute bioavailability studies where you have a single 14C IV microdose together with an unlabeled oral dose. Next slide, please. So I'll just briefly mention this because this was captured already in session 1 by Alan Kennedy and Celine Vaux, but this is an AMS-enabled human ADME design incorporating a microdose. So essentially, this is a 2-period study whereby the first period is an oral standard dose containing less than or equal to 1 microcurie, and then after a suitable washout period, an intravenous microdose containing less than or equal to 1 microcurie 14C is administered together with an oral standard unlabeled dose at Tmax. And from this, one can collect the blood samples are collected, and metabolite profiling, of course, with the IV dose is optional, but blood and excreta samples are collected from both. Many variations of this design are possible. So, for example, the period shown here can also be done in reverse order. Next slide, please. And one of the main advantage of doing a study in this way is that it's possible to build a comprehensive model of drug disposition. Again, I won't spend too much time on this slide, as this was captured in session 1 by Felix and Quinn. But for example, one can calculate fraction absorbed by 2 different methods. One can derive renal blood clearance from the IV data, and then there's various other equations mentioned below that summarize another approach to calculating fraction absorbed that also provides estimates of useful parameters such as hepatic extraction, proportion of drug escaping first pass extraction through the liver, and the proportion of drug escaping first pass extraction through the gut wall. Next slide, please. So just a bit of a future perspective then. So one of the advantages of AMS and the fact that the costs are dropping and more people are investing in this technology is that there's a potential transition to a human-first, human-only approach in DMPK for selected programs. So on the top of this diagram, this flowchart you see here, you see kind of the classic approach where one would begin in phase 1 with a radiolabeled synthesis for preclinical work, so a rodent ADME QWBA, which would enable a dosimetry calculation. And then in phase 2, then this would move on to radiosynthesis and drug product manufacture for a conventional human ADME, so 50 to 200 microcurie dose, let's say, if of course the dosimetry allows it. However, with an AMS-enabled approach, one could potentially skip the animal work since a dosimetry calculation is not required. So in phase 1, one could go straight to radiosynthesis and drug product manufacture and conduct a microtracer human ADME. This could be done in parallel with the phase 1 study, but slightly offset because, of course, one requires sufficient safety data to enable administration to healthy volunteers, or it could even be integrated within the phase 1 first-in-human clinical trial. Of course, there are nice advantages of this. There's potential reduction of animal use and saving of time. You get the key data you need for things like MIST in phase 1, which would enable plenty of time to either synthesize the metabolites and measure them in tox studies using bioanalytical approach, or establish metabolite coverage of the metabolites of interest using the mixed matrix method, which was talked about last week. So that's definitely a potential advantage. Of course, I wouldn't say here that animal ADME studies won't be needed at all, but they would probably be only mainly conducted to answer project-specific questions. Next slide, please. So what are the pros and cons of a human-first, human-only approach? So the pros, of course, is a reduction in animal use. So animal ADME studies would no longer be required in most cases. However, note that the rat Cuba study is still required to support toxicology, so in line with the ICH SC3B guideline. So the con of this potentially is that animal experience from these studies would be unavailable, and this could potentially increase the risk of problems arising during human ADME sample analysis that could compromise the quality of the study. Another advantage maybe is that it provides important quantitative information on the PK of drug elimination pathways early in development, which is useful for modeling and simulation efforts, which is a growing part of the DMPK world now. Of course, it increases the risk that the human ADME, though, is it performed at an incorrect dose or design, but particularly for compounds without dose proportional with time-dependent PK. It allows, having the study early would allow for streamlining of clinical pharmacology plans, so project teams would know early what studies are needed, and this could be compatible with highly accelerated programs, such if you have a compound which is potentially first or best in class and there's a desire within the organization to accelerate development. Of course, the con is that it adds a considerable resource commitment to the phase 1 of drug development when the rate of attrition is still quite high. It's around 50% generally. We get different numbers if you look in the literature, but it's around this value. And the last advantage is that the information relevant to the safety testing of drug metabolite guidelines is known early, allowing plenty of time to address them. Next slide, please. So just before handing over to Greg for the NMR, just a few concluding statements. So AMS is evolving to smaller, more user-friendly implementation with compliant software. So combustion interfaces for sample introduction will likely make graphitization methods obsolete for biomedical applications due to the mainly the increased sample throughput. The current automation possibilities for sample introduction to AMS are likely already sufficient for most biomedical applications, although there is still room for improvement. Runtimes per sample have been reduced to approximately 6 to 20 minutes, which is instrument dependent. AMS availability is still quite limited for biomedical applications. There are a few CROs, a few pharma companies now with their own facility, but this seems to be rapidly changing. Of course, AMS is a key technology that would be required for a move towards human first, human only approach in the DMPK field. So thank you for listening. At this point, I will hand over to Dr. Greg Walker for the NMR part.
There we go. Let's talk about some fluorine NMR and how it applies to human ADME studies. Next slide, please. A brief review of what a human ADME is. I'm hoping that most folks on this call understand this already, but there are two primary endpoints. Mass balance, in which you, conceptually, it's very easy. You give a known amount of compound to a subject and or patient, and then try to account for all of that in the excreta over time. The industry average is about 89% with a range of around 40 to 110%. There is a recent guidance by the FDA that suggests that we should always have greater than 90%, but that's probably a separate topic. The next primary endpoint is metabolic profiling, which is honestly my favorite part of this kind of study, in which you structurally characterize and quantitate all of the metabolites and parents in each of the three matrices that you're interested in: plasma, urine, and feces. Now, all of this is typically done with an analog that has C14 somewhere in it, with an endpoint, a detection endpoint using scintillation counting of one sort or another, and or AMS. So, next slide. So why do they use C14 and how does fluorine NMR stack up against it? I should point out, some people get a little nervous. C14 is certainly a radioactive isotope. N15 is not a radioactive isotope, and that actually lends a lot of ease to the use of it. At any rate, what makes C14 a good candidate for analytical detection in a study like this? It's got all of the properties that you look for in bioanalysis: good linearity, specificity, sensitivity, dynamic range, universal response, which is something we're going to talk a fair amount about because a lot of analytical techniques that people are familiar with don't have universal response and require standards. Also suitability, suitable stability, accuracy, and precision. C14 analysis gets strong check marks in all of those. Now, if you move to the fluorine NMR column, you get strong check marks in all of those except for adequate sensitivity. That is a challenge when using fluorine NMR, and it impacts how you plan your studies and how you execute them and how you prep your samples. And we will talk about that as we move through my presentation. One final point, back one slide. One final point, and I hate to state the obvious, but it is. Your compound for a radiolabel study must contain C14, and for a F19 study, it must contain a fluorine of one sort, and they both need to be metabolically stable. Now we can move to the next slide. In terms of analytical equipment for a fluorine human ADME study, the necessary equipment, almost all of it is already in your labs. Balances, homogenizers, mass spectrometers, incubators, just pipettes, normal lab equipment. Now, what is probably not available in your lab is an NMR system. And it's not just any NMR system. If you've been paying attention to the NMR world, there's a new push toward benchtop NMRs, which are much smaller and much less expensive, but they don't have the sensitivity or the dispersion in order to do a study like this. You need a high field instrument, something 600 megahertz or greater. And even that isn't necessarily sufficient for this kind of study. An integral part of an NMR spectrometer is its probe. That's the part that the sample sits in once it's inside the magnet. And your probe has to be designed for 19F detection. There's lots of different varieties of probes, and you need one that is designed for fluorine detection. Now, one last point before we move on, and this is something we're going to talk about throughout my portion of the presentation, is that your successful human ADME study with an F19 NMR detection scheme, it requires a lot of careful consideration, both your NMR equipment that's used, which we've just talked about, the study design, and the pharmacokinetic behavior of your molecule as well as the analytical behavior of your molecule. If you're experienced at all with proton NMR and quantitation using proton NMR, you have a selection of different protons or hydrogens to quantitate from. If one is ill-behaved in one way or another, you can move on to the next one. That's not typically the case with fluorine NMR. You typically have a single fluorine or maybe a couple fluorines in your molecule, and you're limited because of that. Next slide. A little bit of analytical view on fluorine NMR. Fluorine NMR is extremely linear and has a wide dynamic range. The dynamic range is typically limited by solubility. When things start falling out of solution because of solubility issues, that defines your upper limit. On the left-hand side, we have a nice concentration response curve, good linearity, all those sorts of things that you'd want. This comes from a compound that has a single aliphatic fluorine on it. On the right-hand side, we have 3 NMR spectra, 3 fluorine spectra that demonstrate sensitivity, specificity, and background. The blank on the bottom, you can see that there is virtually nothing in it. Whenever I make these presentations, I always get questions about fluoride from drinking water and PFAS and that sort of thing. Very seldom do we see anything in the blank at all. The next one up is certainly the detection limit for this particular compound, which is 4 micromolar with a signal noise of about 6.2. And then the final one, so that you can actually see something, is 100 micromolar standard. Nice sharp peak, nice sharp resonance. Next slide. One of the things that people have a difficulty with in terms of NMR is sort of really getting a grasp around the universal response. I think that's because most people start out their careers using LC-MS or LC-UV, in which standards are required to quantitate any analyte that you might have. But that's not the case with fluorine NMR. In order to demonstrate this, I went through the Sigma catalog, and I find the compound that has 6 fluorines that have the same chemical shift. I did a nice standard curve on it, just like you saw on the last slide. A great response, nice and linear, just what everybody wants. Now, if we have universal response, what we're going to, if we lose half of those, if we go from 2 methyls down to 1 methyl, you'd expect the slope to be about half of that. So I did the same thing with fluoxetine, and indeed you get half the slope that you had with compound number 1. And just to really press it, I created a standard curve with linazolid, next one. And again, linazolid had a standard curve, or had a slope about 1/3 that of fluoxetine. So if you normalize all of these points to the numbers of fluorines, next slide, you get an incredibly straight line. These are three separate weighings, three separate preparations with multiple samples in each line, and you end up with a straight line. One more slide. With a really high correlation that is independent of the chemical motif that you're analyzing. Now, what this means is that you can use any standard that you want to quantitate against your compound in your human ADME study. And that's a real advantage. All right, next slide. Just to sum up some of the pros and cons of fluorine NMR, we're going to do this a couple of times so everybody remembers it. We have excellent linearity and dynamic range. We have universal response, which is unique amongst many spectroscopic techniques. We have a high degree of specificity. Our stability, accuracy, and precision are all good. And we have a very low background with almost no endogenous fluorines. We do occasionally see a contaminant, but it's not in the range of the PFASs or of fluorine NMR or of fluoride from drinking water. Now, 2 points that I haven't brought up yet is that fluorine NMR is non-destructive, which is really a plus. You get your samples back. And also, it has a very wide spectral width, which in many studies we've taken advantage of. The spectral width is roughly 250 ppm. So if you have contamination, you can usually isolate your resonance of interest away from it. The cons are its sensitivity, which then leads to more involved sample workup. And it is an offline analysis. You have to collect fractions and reanalyze them. Some people don't like that, but you also do that with AMS as well. So it's a disadvantage, but it's one that I think we can all live with. Next slide. All right, the next 2 slides are going to be about workup in terms of how comparing a C14 classic human ADME study or ADME study with a fluorine-based ADME study. And on the left-hand side, we have sort of an outline of the C14, and on the right-hand side, we have an outline of the fluorine. I'm just going to highlight some of the differences that we have. Obviously, you have to have radioactivity if you're doing a C14, which means it has to be synthesized, which is expensive and time-consuming, all that sort of thing. One of the big differences is that with the C14 study, you will do your total C14 in plasma and determine your plasma pooling strategy based around that. We have not had sufficient sensitivity in terms of fluorine NMR to do that individually with the samples that we have had. So we base our pooling strategy around the pharmacokinetic behavior of the parent and the metabolites and previous experience with the molecule. Once you get past that, you pool your samples. On the C14 side, you're pooling 1 to 5 mils. On the NMR side, you're pooling somewhere between 20 and 40 mils. After that, things are largely the same, except for the fact that on the NMR side, we cannot determine processing recovery in the same way that you do with C14. I mean, you are very sure of what your recovery is in C14. With fluorine NMR, you have to rely on standard additions and then recovery of those from parent or synthetic metabolites that you may have available. And in either case, when you get to the bottom, the end result looks the same. You can't tell the difference between the chromatogram, the reconstructed fluorine chromatogram on the right, or the reconstructed C14 chromatogram on the left if you just ignore the y-axis. Next slide. A very similar kind of slide for the excreta. We do determine the total amount of fluorine-related material for both studies, C14 and the and the fluorine NMR, which then drives your cooling strategies for your urine and your feces. And again, we have the same difficulty of not being able to provide the same recovery assurance that you do with C14 with fluorine NMR. You have to, again, rely on standard addition type experiments. Again, the volume is different for the two in terms of sample volume. You may on C14 side for urine may use anywhere from 1/10 of a mil to a mil. It varies depending on the sensitivity need that you have in the fluorine side. But again, in the end, you end up with two sets of chromatograms that are equivalent, give you the same information, give you the same data. All right, next slide. Talk a little bit about the clinical comparison. On the C14 side, this requires C14 synthesis, which is sometimes complicated and certainly expensive and takes a while. From the clinical point of view, this is typically done on a single dose and not at steady state. Steady state is the preferred protocol to address missed concerns, and that's typically not done in C14 studies. Fluorine NMR studies, the API obviously requires no C14, so you just use your drug. The excreta, because of the freedom of not having any radio label or not needing any radio label, you can do it in an FIH SAD leg or in a MAD leg if you want steady state. And another really positive attribute about this is you can use the normal formulation rather than trying to design a specific formulation that takes care of your C14 dosing. Next slide. Critical clinical study points. These are often the same for both C14 and for N15, 19F. You have to capture the appropriate points for all of your matrices, plasma, urine, and feces. You need that for both of them. Metabolite abundance can complicate things. It's a lot easier to do a study that has a single major metabolite than 10 minor metabolites. That's a lot harder. It's hard for both techniques, but I think it's actually a real challenge if you're doing fluorine NMR because of the sensitivity. The volume of source material versus sensitivity is something that you have to consider. If your lower limit of quantitation for an NMR, the NMR system that you have is 500 nanomole, and I've just picked that as an example. You have to back calculate to the volume of plasma or urine or fecal extract that you need. And sometimes this can be prohibitive if you don't have sufficient substrate to end up with 500 nanomoles in a single tube. Plasma calculations should be based on half-life and CAV. It's all about exposure. If you get the right compound with the right amount of exposure, you can do a surprisingly low dose. Excreta calculations are based on drug-related material that is excreted in a per unit time, usually a day, and your extraction efficiencies. The last point is to be careful with co-medications. This is not necessarily a problem all of the time, but in certain clinical settings, there may be subjects or patients that are co-medicated with things that have fluorine. This is when your wide spectral width may help, depending on the compound that they are being medicated with. It may not show up in the same spectral range as your compound of interest that you're doing a human ADME study on. Next slide. One last analytical point. This is some real NMR data. This is not made up. This is a human liver microsome incubation, just to sort of prove a point about the utility of fluorine NMR. And again, if you look at the reconstructed chromatogram on the left. It looks exactly like a C14 reconstructed chromatogram that you might have. Now, one of the differences is that that peak that's been highlighted at 20 minutes contains 2 co-eluting metabolites. If you're running this by AMS or if you're running this by the classic C14 method, you either have to accept that you don't know what the ratio is of those 2 metabolites, or you have to go back and redevelop your chromatography. However, if you're using 19F as your detection scheme, you can query the individual points on the right-hand side. You can see the individual NMR spectra that make up that peak. You can sum the individual resonances and come up with the contribution of each different metabolite. And you can see that the one with the earlier chemical shift has 14.3%, and the later one has 3.6%. And if you're doing your math carefully, you're going, wait a minute, that doesn't add up to 19%. But if you look in the upper right-hand corner, there's a couple of very small metabolites that have resonances up there that add in your additional 2%. So you do have total recovery of the amount of C14 that you saw in the slide or in the panel on the left. All right, next slide. And one of the real advantages of using fluorine NMR is that it is a non-consumptive technique. Your sample, when the NMR spectroscopist is done with it, when he's done, or he or she is done quantitating the fluorine, you can run other NMR experiments. On the right-hand side, this is a subsection of an NMR experiment called a TOXI, which helps you understand proton-proton connectivity. And when the NMR person is done with it, they can give it back to the drug metabolism scientist or a mass spectroscopist, and they can continue to do further experiments because you have an isolated sample of known structure and known concentration that they can work with further. All right, next slide. In summary, analytical pros for fluorine NMR, linearity, all of these things that we've been talking about, dynamic range, universal response, non-destructive, wide spectral width, all of those things. Analytical cons, the sensitivity, the more involved sample workup. Of course, obviously, you have to have a fluorine NMR or fluorine in your molecule. And additionally, you do have to rely on standard additions to calculate any recovery. From a clinical point of view, the pros requires no C14 synthesis, which adds a great deal of cost savings, time savings, and flexibility for you. Your excreta can be done in FIH SAD studies. Your plasma can be done in MAD studies so that you achieve steady state, which actually is a big issue. The cons, you have to have sufficient exposure, and you need to work with your NMR experts in order to define what that is for your specific molecule, their specific instrument, and your program. And I think the last slide, oh no, one more slide, 2 more slides. The future. We've talked a lot about some of the failings of NMR or some of the shortcomings, largely centered around sensitivity. There are things that we are doing in our lab right now that involve hardware and software and experimental design to increase our sensitivity and increase our throughput. We're not prepared to talk about those right now, but there are things on the forefront that will make this a lot easier. Improved sample workup procedures so that we can not worry about absolute recovery. We're thinking about different NMR approaches that we might have, or maybe even bringing in some other analytical techniques that will help us with that as well. Now the final slide. You don't do this sort of thing alone. Scott and Loretta and Raman have been absolutely integral in all of this, and I am proud to say that I work with them. The rest of our group, every one of them has helped in one way or another with the information and the data that you've seen in this talk today. And I'd like to thank them. I'd like to thank the IQ group for allowing me to be a part of it. And I'd like to thank you all for your time.
Great. Thank you so much. Thanks to all the presenters. This was wonderful. We have few minutes for Q&A. You can use the functionality for asking your questions, and Jason and I will rotate in asking them from the speakers. I'll go first. Maybe this is actually to everyone. The question is, are there any known excipients that interferes with quantitative methods summarized in this webinar? You know, fluorine NMR, Greg talked about AMS, and Philip talked about ICP and others. Greg, you want to go first since
if anything that has a fluorine in it, but again, you may be able to work around that if your compound of interest has a chemical shift that's significantly different than the excipient that you're worried about.
Alex, you want to handle the AMS?
It's unlikely that excipients would have any impact at all on the measurement.
And Philip?
Yeah, for the icp-ms, for the bromine work, for instance, we also did some laser ablation work as a alternative for QWBA, and then we, for instance, saw that the tape where the cuts were made, let's say, was also full of bromines. We had to make thicker cuts, let's say, to get rid of the bromine background. So that's an example where, of course, yeah, you will see these kind of elements also popping up in excipients, and also chlorine. Of course, our body is full of sodium chloride, magnesium chloride, so that's also full of background. So you have to, it's of course easier, it's inorganic, you can wash it away in your LC system, but background, of course, it's a bigger concern than with radiocarbon, for instance, where it's not a concern at all, where you have the selectivity. I always have the selectivity.
Okay. Another question. This probably goes to Alex, but I think all three could address it. But how likely is it for the FDA to approve the use of the first human only ADME in drug development, and what are the roadblocks to this paradigm?
Oh, that's a good question. I mean, I don't see why they wouldn't, right? I think the human ADME will help you to address missed issues. It will, and of course, it doesn't stop there. You will still need to do other clinical pharmacology studies like DDI and so on once you know what the metabolic elimination pathways are. But you still need a rat cuba study. This is needed in support of toxicology. There's a guideline on this, so you can't completely escape animal work. But I think many of the old paradigm of doing an ADME study in every animal species, you know, like a rat, dog, a rabbit, mouse, could go away. I think it already is to a certain extent. I think many companies do a rat ADME anyway, but you see less and less the full suite of species being done. So I don't see why the FDA would go against it. I mean, I think there still is a place for ADME studies as well. There could be specific questions that could only be answered this way. But yeah, that's my perspective. I don't know if anyone else has any comments.
And probably the biggest concerns were already addressed during the presentation, also that you need to be in the linear range or have the right dose in mind already, and also the final formulation, which is not always set at that stage. But yeah, that was already addressed during the presentation. Yeah.
Mm-hmm.
Next question may also be addressed by everyone. Maybe I pose it to Philip first. For new techniques, such as ICP in this application, how do you see the regulatory uptake? What steps would you take to have it approved for a particular molecule?
Yeah, so we had these lunch and learn sessions before. The same webinars were given, let's say, as lunch and learn to the FDA, and there it was actually also addressed this question to the FDA, and they were quite open to new technologies. That's at least the answer we got. So that's positive to hear. And also, when we did the icp-ms work back in the, I think it's 10 years ago, something like that, FDA was quite open to the results and the technology. With EMA, we had some discussion and finally also accepted. I think it's just a matter of explaining the data and how the technology works, and then you can convince them, I think.
If I could add, I think it helps to have some background in publications and peer-reviewed journals. You know, certainly the fluorine NMR, when we began working with it, there was already a set of data that was out there, and that was very helpful.
How about I have a quick question for Greg. So you talked about you mentioned in your presentation that the the NMR and the AMS both required fractionation. I was curious, where does LC NMR fit into this situation, or does it not?
I would say it does not. I spent about 10 years working with LC NMR, and it was 10 years of frustration. Once we got cryoprobes and better understanding of sensitivity in terms of those cryoprobes and better sample processing, it's just hard to have the same time of acquisition in a flowing system. So you know, there are certain applications where people use LC NMR at this point, but I think they're becoming extremely small.
Great. Thank you, Greg. This could be the final, just eye on time. Alex, similar questions as the online AMS. It is in the literature that allows kind of mapping metabolites, AMS and mass spec. Have you had that experience, and you know, is your experience similar to Greg?
Sorry, in what sense? You mean correlating the peaks in AMS to the mass spec data?
Correct. But having it to be online. I think Livermore.
You can't do that. I think online AMS, direct introduction into the AMS system with the LC flow. I'm not aware of a commercially available instrument that can do that very well at the moment. So I think what typically happens is you do an LC-MS run, and you have a post-column split. It goes to a fraction collector. And then the fractions are analyzed by AMS, and then you can correlate this with the LC-MS data.
So from the fluorine, I'm sorry, are you done, Alex?
Oh, yeah, go ahead, Greg. Yeah.
So from the fluorine NMR point of view, we do much the same thing. We isolate with a splitter. We have an understanding of what the molecular weight is. But then once we're done, we take the samples that we have that contain the fluorine, and we reanalyze them by mass spec to make sure we have the same data and the same profile that we thought we had.
Great, thank you. Apologize to those who the questions we couldn't get to, but you could have them go to Maya, and we would try to address them as best as possible. So the concluding remarks. So this was the third session of Human ADME. I would like to extend our heartfelt gratitude to all the speakers for their insightful presentation, and to all the members for their active participation and collaborative spirit. We look forward to 2 manuscripts that are scheduled to be completed in the coming months. These will capture the critical findings and discussions in our sessions. I'd like to thank my co-chair, Jason Bohr, and Maya Leah Marshall from IQ. Thanks again, and we look forward to your participation in future endeavor. With that, we close this session. Thank you.
Thank you all.
Thank you.
Thank you, everyone.